Yeah, so I'm noticing something a little bit goofy about the slide order that I have in here now. Let's leave it like that.
Do you need some help?
No, I think I put the slide in yesterday. Then I only added in the slides 2. But it's okay. I won't. I'll figure it out.
Okay. Well, if you need help, IM me.
I'm glad to see Spiff here. And Spiff's YouTube is now live. So Spiff, just a quick warning, I'm going to have a change in camera angle at the end. I'll be demonstrating something. It'll be up by the upper left into the trees. No problem.
Oh, I forgot about that.
Oh, and I'll go ahead and set up the transcript as well.
Okay. I have a statement about recording. Okay.
Ooh. You know, I only get 30 minutes of transcription per conversation. Interesting.
Oh.
Yeah. But what I can do is I can take a quick pause and make a new one. Let's see here.
Just reload. Yeah.
Okay, I'm going to put this link in the local chat, but only good till 1:25. But if I can, I'll refresh it really quick.
Okay, I notice here I'm going to say that I will put the stream into local chat. I'm not sure I have that.
Brila, no, I'll have to refresh the conversation. I'll have to make a new link, I think.
I was thinking of the YouTube stream. Okay, that's all right. You have that, Brayla? Okay. You want to insert that in the chat at the right time? So that's how you spell that word. Yeah, I know. We'll wait just a couple more minutes. People to arrive. Yes, the presentation is in voice.
Yeah, I just see a few people that don't have above their heads, so I wanted to.
I see. Okay. I can put my text into local. This part of it.
We'll also ask them if they need text.
Okay.
I can do that. It's fine.
Okay.
Oop. Okay.
Okay. Thank you, Brayla. I forgot about those.
Yeah, the Google Doc link will just be for later.
Okay. My fancy clock says it's 1 o'clock, right on the dot. So to give Stephen plenty of time to present, I'm going to go ahead and begin. And we will have this transcribed to text for anybody who prefers to see it that way. Let me extend my greetings to you. We have a nice Irish setting here, and it took us forever to find this green carpet, of course. But at any rate, hello everybody. My name is Meryl McBride, and I have the pleasure of serving as your host today. I want to welcome you to this installment of our VWEC Expert Series, in which we ask educational professionals to share with us their experiences and their insights into ways that virtual worlds can be used to engage students. I am pleased to introduce today's guest presenter, Stephen Gager, who will share with us how he used social virtual worlds to teach biology and genetics at the college level. As you will discover, he wanted to do more than simply deliver a lecture class. Stephen is currently a researcher at Corteva AgriSciences. Earlier in his career, he taught biology at the University of New Orleans and at Ball State University. In each case, he leveraged the potential of virtual worlds such as SL and OpenSims to give additional life to the students. Stephen has from the University of Chicago. Stephen has a long and distinguished career working in Second Life and OpenSims, is actively presenting in Science Circle, and is providing talks to various science interest groups. We will be using Otter AI for Stephen's talk. In addition, please know that this presentation will be recorded and streamed on YouTube. You can find the stream at the address that we will insert into local chat. feel free to questions to chat during his brief talk, and I will collect them for him to answer before we go to the back of the venue. All right, Stephen, that's all yours.
Thank you, Merrill. Thank you to the VWEC for extending an invitation to me to talk about the work I did in Second Life, teaching college biology. Very appropriate for Merrill to be my hostess, that she was the one who sponsored the grant program that got me at the very first workshop where I first rezzed, and then things went from there. So a little bit of background about myself very quickly. As mentioned, I was a postdoctoral researcher before I became a teacher at University of New Orleans. And I was not an educator, and I never got trained in education. But I will say that I tried to do stuff partly to just push the envelope of what I thought was interesting, which is why I went to the workshop in the first place. And I think that Second Life has always represented educators, that type of platform opportunity to enhance educational experiences. But I never had any formal education training. And so I kind of wung it in just trying to do what I thought was interesting and enjoyable and something, but keeping in mind something accessible and fitting into the curriculum of the class. In quick summary, the classes that I taught, and this was at University of New Orleans, Department of Biology, were what was called contemporary biology. And this was a class that was meant for non-biology majors. And in some ways, the teaching level is a little bit more accessible. You aren't necessarily trying to get these students to remember and memorize things about Mendel and genetics that they'll use in their life. And so that's a little bit easier in certain ways. But on the other hand, I think it was also more incumbent upon those types of non-majors classes for the educator, for the teacher, to make sure they impart a very important impression that the students would have for the rest of their life. And that was one reason that I really wanted to teach this particular class as an online class, was to make sure that the combination of the platform, as well as hopefully the interesting classwork in it, was something that they could remember and take with them and appreciate biology, even though they were not going to that particular field. So the classes were a summer online class in 2010, also the next year in 2011, and then the spring of 2012. And that was the extent to the times I taught the class. A lot of what I'm going to talk about here largely pertains to that third iteration, because as you might imagine, the first iteration looked very different than the last one. One thing I also did, which not every Second Life educator attempts to do, and this is perfectly reasonable, is to have as possible of the assessments be an in-world activity, something where they're building, having an assessment, and actually have that be the graded part. But one thing to keep in mind that the university at the time preferred to always have some in-person component where the actual person had to go to a proctored exam where their identification could be confirmed. And I basically said, well, let's have a final comprehensive exam, something where they had to have paid attention for the whole class. But that was actually an in-person exam, in terms of how that assessment in world versus an actual paper assignment. I will say also that the University of New Orleans, a lot like other places at the time, did not do anything to generally bring their students into the world of Second Life. There weren't student workshops, there weren't, use SL or learn how to use online platforms for classes. And so the student body that I had to teach were people that at the same time I was teaching the class, I was almost always teaching them how to use Second Life, which of course is a major challenge in that type of way. To any institution wanting to move more distinctly into Second Life, or any online platform, that they do a diligent job of making sure that that's accessible for everyone. Now, overall, even though I wanted to do a lot of really interesting things that advantage of Second Life. The basic format I wanted to keep straightforward and familiar. And so what you'll see here is a snapshot of my classroom. I used the Clever Zebra auditorium. I used the Clever Zebra lecture, and I presented slides. We also had available as PDF from the Blackboard management system. And so in many ways, that was a very familiar experience. One thing I'll say, and this is one of these things of the learning curve that I had, was it took me a while to always remember and keep track of the 20-meter local chat, because I encouraged the students to have local chat going on during lecture and to use that to ask questions or to talk to each other. It wasn't I learned to say, as an example of those things that you learn as an educator, the technical aspects that, oh, I should make sure one by putting out markers for what 20 meters was. This snapshot, he was a student at the time. He actually wrote on 2 SL classroom experiences, one of which was mine. So he sat in during that class, and his thesis is available on the web. And I'll have a batch of material, or if you want to ask me about it, I can get it for you. But here's the citation. So I think students in general adapted to that type of opportunity. They came to class consistently. They were able to absorb and learn the material as you would maybe any other normal class. What I also used the space for, and this is a snapshot from outside the auditorium, is I set up a whole bunch of media boards. And so the textbook material included a whole host of MP4s, various videos that were correlated with the class material. And I hosted those on my own Mac OS X server using the Darwin X server, and made those available for the students to do and to look at while they were in world, as compared to it necessarily being something they did outside, although they were also available on Blackboard. Trying to, again, enhance and create this multimedia experience above just having them come into the classroom. All right, so let's talk a little bit about the teaching and how I tried to use Second Life. There was one component of the class that was to give them a flavor of biology, an opportunity to talk about biology and learn some biology, but they were not graded assessments. Second Life offered a lot of great opportunities along those lines. My favorite would be the Voyage of the Beagle, which the Nature Publishing Group hosted for a while. And that was something that you could do a gameplay, even just absorb a bunch of different material that was about Darwin and the origin of species. One thing I did as a participation event was to go to Science Friday. One thing I purposely did was I scheduled my lectures to finish when Science Friday was going to start. And so I'd say, hey, class is done. We can go sit in on Science Friday. We had our own little group chat sessions, and a lot of times had a really good time just chatting about what was going on there. Again, something else that's also not available anymore. And of course, Genome Island is a wonderful place where there's lots of great information as well as interactive activities. But again, I did not set these up as assignments until I felt comfortable enough where I could rewrite the science and the worksheets in a way that suited my students from Genome Island. So in that third class, those did end up becoming assignments. And then what I'm going to talk about today are 5 different activities that I developed or co-developed specifically for and designed for this classroom. So one of the reminders about Second Life and some examples, Voyage of the Beagle was such a great experience, and I know they put a lot of money and dedicated effort, and it's so great, but it's not available anymore. And this is, I guess I captured this as a screen capture just to make myself sad on occasion, I don't know. But here's a picture of Genome Island. And what was really nice about it is that's well laid out. It has markers to get to different places, and it included in note cards the lesson plans, the activities that you had from each station. But of course, that would end up being one of the challenges, would be that if I wanted to lead a tour of the students, how did I get them there? How did I manage to teach them how to get out of the classroom and actually navigate, and then also navigate together as a field trip? Another thing that I did and actually a way in which I met many of the people in the audience here, was I set up some animal poster boards. So this was the kind of capstone in-world assignment where the students would pick some animal, and they could pick whatever they wanted to, and then basically write about its characteristics, its habits, its ecology, anything interesting about its history, what its geographic range is or was. But they all got extra credit if up or use Second Life objects to represent that animal in world. And so you'll see here, those are actual avatars of a white tiger, a crocodile, and in fact, they did a vulture, but they got the raven avatar, but they have some vultures flying in the background. So those are the actual students, not just objects representing the animals from the poster board. And so the assignment also was I asked people from the community to help grade and walk through the students in the poster board. Again, one thing I'll point out a little bit later, we had a lot of space available on our university islands because we had basically easily 6 contiguous lands that I could take advantage of for the building. Otherwise, having students walk around and have voice or chat conversations with visitors would have otherwise been probably uncontrolled chaos. But this was an assignment that I think a lot of students really enjoyed, that they could be an animal. So in terms of getting to the actual assignments that were credit and the students were expected to perform in Second Life, I had a couple design principles. And the main thing here is to make sure that because we're using Second Life, which is this programmable, 3D, interactive environment, do something that actually takes advantage of that environment. And how could that be best leveraged in order to enhance the learning of the concepts? And so I'm breaking this down into 4 different items, which is, it's really nice to have 3D objects that that are spatial and interactive to teach about spatial concepts. Another thing that was popular at the time was this idea of different learning styles, and that some students prefer certain learning styles, but also employing multiple learning styles can help reinforce the material. So actually doing building, which was a type of kinetic learning, would be a way to help students learn the assessment. And then interactivity, again, that's a key part of Second Life and the environment. And from a science point of view and the ability to have this programmable space, how can we generate novel data? How can the students really feel like they're owning their data and have it be generated by the in-world objects? Again, these aren't design principles that happen for every single assignment, but something that I wanted to have at least one for every assignment that was done in biology. And I think it's an important thing that if you want to distinguish your platform that you're using for some particular educational purpose, that your assignments also reflect a way that justifies using the platform. And at least for biology, this was something I thought were real added value. So to go into more detail about some of these design principles, when it came to teaching about spatial concepts, like I said, if you can have spatial objects, maybe you can teach those spatial concepts a little more coherently and hopefully get better learning outcomes. Now, the downside of doing these things is this extra technical hurdle of camera and avatar placement, that if you have a spatial object, well, what perspective are you looking at? How are you viewing it? Is it your camera? Is it your avatar? Do you have to use mouse view? And also very important is understanding build permissions and the ability to install stuff. And basically, how are you getting these out into the world for the students to see? When it comes time to actually the connecting and interacting with these objects, again, you can engage their minds kinetically. And it actually, one thing that the students can't do, they're going to have to learn the material to be able to do the assignment, because they can't just copy cheat if you're doing something that they basically have their hands on. Now, of course, the downside of this teaching is that I teach them rudimentary building skills. So there were whole class times that were dedicated to having to teach them how to do that, even at a very basic level of just moving things around. Now, I did have great help from a friend of mine who was going to be, just on her own personal volunteer time, a TA that would help students with that, even though she didn't know biology. And the downside is that if you make assignments where they can't copy sheet, you have to also make sure you're individualizing And so that means you can't just grade off the key. You can't just have one simple thing of what's right or wrong. You actually basically have to grade each individual assignment, which was fine for my class size. Like I said, I was generally under 25 students. And of course, one of the key things, space is at a premium, and you need sandbox space. And let me tell you, once you start having students build and res stuff, you have to have that bulk return power for it to work really well over time. So let me show some of the two main objects I did for the chemistry. So one of the learning lessons was just understanding basic chemistry. And in terms of chemistry, some basic concepts behind that are valence shells and how electrons bind atoms and elements together. And in fact, there's a lot of just very simple math and problem solving you can do in order to have them solve these questions in an assessment. But what you're really getting down to from, again, contemporary biology, basic chemistry point of view is, why is it NH3, right? There's an actual reason why it's NH3. That's ammonia, by the way. Or why is it CO2? Why can't it be CO3? And then, for example, hydrogen cyanide, H, C, triple bond to N. Like, how is that chemistry being determined? And that way they can understand a few basic chemistry concepts, because some of the key chemistry we're building up to is understanding how animals capture energy from sugars and also how plants make sugar, because that was one of the basic foundational ecology principles from a basic contemporary biology type of class. And so here are some screen captures of the objects that I built. These were basically simple resers where when they would click different items, it would push them out into space. It would res a fresh object, and then they could try and move that around with the simple editing tools. And I have a question here. It's like, why did it take me so many design iterations? Like, one simple thing that you might notice on the left-hand side, there's a big green space underneath the element names. And that was so when they res the actual atoms, that they had a backdrop that contrasted with the objects they were trying to work with. It took me a while to actually try to figure out some of these basic concepts for building. And so in terms of assessments, here's an example of one where you might say, how would you make a molecule with 4 hydrogens, 1 carbon, and 1 oxygen? And what's relevant here is that there's really only one way to build this where you satisfy all of the basic chemistry of how these shells go together. And so that's what I'm actually showing. I'm giving you the answer to this one, is that you'll notice, and this is one of the key concepts of the chemistry, is that when you count the number of yellow dots that surround the carbon, and you count the yellow dots that surround the oxygen, there's a total of 8. And what's happening is they're sharing 2. And in each case, they're also sharing electrons with a hydrogen in that conformation. And so what I'm going to do here real quick is right here in front of me on the ground, I'm going to res, or even just block my own view. This is an example of this element reser that, again, if you want, please do not click on it, but I will demonstrate. If I click on the magnesium, it would pop this object out, and then basically students could manipulate these to make new molecules. So again, this was actually a force-facing type of object, which is why that one resed out in a weird spot. These are available. I just basically made these. It took me a long time to make the images and do stuff with them. If anybody wants a copy, I'm happy to share that. But these were the types of interactive objects that I wanted to build for students in order to help teach the basic concepts of chemistry. One of the other concepts in chemistry was not just that, how does the chemistry work, but this is a biology class. And so we're trying to understand how organisms utilize that chemistry to make energy, understand how, for example, plants can use photosynthesis to take simple molecules and turn those into complex energy-containing molecules. And so what I have here is an example of something that's used for assessments, which is the Calvin cycle, which is how plants, the chemistry reaction that's important for taking carbon, and that's the black 2 red balls you see at the back there, that gets incorporated into a cycle in which the other carbon-containing molecules, again, I didn't ask them to make the whole molecule. I just wanted them to count the number of carbons that were relevant reaction. And I basically asked the students to build these on top of the platform. That was their assessment, was if I gave them a chemical reaction, could they build that? And I let them do it as teams. Can they collaboratively build this to understand what the carbon makeup of different steps was for each one of those? All right. Now, one thing I'll just mention here quickly is that there's this great tool that someone for the American Chemical Society made, and it was Heroes Chemistry Resor. And this looked so cool when I first saw it. I'm like, oh wow, you can take any chemical, go to 3D data bank on the web, download a file, and then this reses the actual chemical structure of this in Second Life. And it's really cool. I have all the amino acids, and I use this as like class demonstrations, but I usually just use them as a teaching tool because there was not a way to make them a manipulative that I thought was worthwhile for this level of technicality know-how for Second Life for the students. Like, you could imagine having these moieties and other stuff, being able to, can you combine these and put these together? But for a contemporary biology class, I did not think it was relevant as some sort of teaching assignment, and it would just be too challenging to get them to work. So those are my fun chemistry manipulatives. The next level of stuff that I tried to make and assign to the students was to really leverage the interactivity. And the big advantage for interactivity is a deeper learning engagement. But you'll notice the list of downsides was a lot larger, right? Again, how, if you have something where they're interacting, how are you placing them? How are you getting them to interact? How is the camera angles working? You know, you need land space and a lot of object impact numbers to be able to have these work as installations, or for the students to interact with them. Also traveling. That again, if you can't have this be in the classroom. You have to have the avatars get someplace in world. And so that was also a challenge. And of course, a lot of this required scripting skills. I was not a scripter. I'm not a programmer. I didn't know how to do any of this. I took some copy paste things and manipulated them as I needed to, but you'll see some of the more interactive stuff. I basically relied upon a volunteer scripter collaborator named Kira Komarov, who basically did some really complicated scripting for me. And I'll share some of that in a bit. Some of this required purchasing. And again, one thing usually is a story that is how much bunny food he ended up purchasing for my class. And of course, the generating novel data, you know, again, this is one of these things where, especially in a science class, you want to give them this experience of analyzing data. And it's not just a data set. It's something where you can basically let them feel a little bit of ownership, where the data that they get is something they need to analyze. And they can't just copy and paste from somebody else. They would actually need to go in and interact with material, understand it. Again, they might ask someone else for help in order to be able to get help with understanding and interpreting it, but the actual math and other stuff that they had to do was actual ownership of their data. Okay, and what I'm going to do is post really quick in a new Otter AI transcript link, which should take us through to the end of the talk. If there are any problems with that transcript, it's working, make sure that it's shareable. All right, thanks. Okay, so, but again, while they can't copy cheat and it really engage, of course, at the same time, as a professor, you just don't have a grading key. You have to be able to grade and redo the math of what they did for their assignment. So you can't just grade off of a template key. Now, one thing we're going to have to do real quick is go through a little bit of genetics so you can understand what the learning outcomes needed to be and what the topic was. And some images from the Campbell textbook that I used to have. And so trying to use that to represent how the class worked. So basic biology always includes basic Mendelian genetics. I'm sure all of you have been exposed or heard about it someplace or another. But the key part about it, when we think about sexually reproducing organisms, such as ourselves, that when it comes time to make a new organism, you get half of the DNA from the sperm, from the paternal line, and half the DNA from the egg, from the maternal contribution. But when it comes time to understanding any given gene, there's just this basic math behind it. That if there's a 50-50 chance of one of two things coming from the sperm and 1 in 50 or 50-50 chance of something coming from the egg, then there are 4 different possibilities. And at least in most cases, if there's only 2 choices from each, basically there's a total of 4 choices that are represented here. This is your standard Punnett square. It's very basic math. And so the challenge is not to teach them probability. The challenge is to help them understand how this relates to biology. And so Mendelian genetics, Mendel had these pea plants. And so the classic way of teaching this is to basically talk about the same things that Mendel looked at when he did his work. And so flower color was something, again, the peas he worked with had flowers. And there was this basic concept of one version of the gene is dominant to the other. And so the inheritance pattern that Mendel worked with was that if you had what are called true breeding plants, ones that were true breeding for purple, and he mated them with ones that were true breeding for white, again, interacting with each other, self-fertilizing, always the same color, then in that F1 generation, they were always purple. And that indicates purple was dominant over white. And then in that F2 generation, if you let that F1 generation breed with itself, then what became uncovered was that 1/4 of the time you got white back. And so that meant that white and these genes were discrete elements that existed, but they were just being masked by the dominant phenotype. And now when we go back and if you focus on the slide at the very bottom, we talk about those 4 different possibilities of, pollen and egg in this case. There are your 4 possibilities, but what you see are 3 of them being the the cryptozoology of the species as if they were a predator. But that's not the best part. The best part is the actual population genetics simulator that I built. And what this was, it was user-defined parameters that you can vary and represents this Hardy-Weinberg equilibrium as well as selective pressures. And so the key thing here, and what I really loved the most about this, was that we basically used Kira programmed in, and actually what I'm going to do quick is her explanation of how this actually worked and put it in local chat. Amazing scripter. That this was building random data. So again, first of all, the great part was that you had this, you could define the P and Q values, the frequency of alleles, but then also change the number of moths, as well as change the number of birds that were predating on the moths. But these were also built to have a random placement of the moths and the birds on top of this disk. And using the generator, the birds would fly around, and then collisions would cause a moth to die. But the collision death was also based on the probability due to the coloration of the moth on the background. And so, for example, here, in the image I have shown here, if a bird collided with a moth, there was always an equal chance, whether they were brown or black, whether they would die and then get recycled into the allele pool. But if the background was set to brown, then the brown had a higher chance of survival than the black. And conversely, the black had a higher chance of survival than the white. And so I'm going to show the worksheet that I built for this, that I just built a basic assessment where I asked the students to res a certain number of moths, look over time at what was happening, and basically generate their own data, which would be random for each student. And that allowed them to basically build their own data sets for them to interpret and then, of course, remediate a grade. And so in many ways, you know, this ended up being some pretty interesting assignments that basically just asked some basic questions about whether they understood the power of the interaction, how the interaction was working, and if they could understand this basic concept of camouflage or crypsis, in order that basically changed how their survival would work within a population that was changing over time. Okay, so what I've done here if you look up to your left, I have this population genetics simulator here. And what I'm going to do really quickly is tell it to run. And you'll see some local chat come in. Again, this is an interesting aspect of it, is that we had to have local chat so the students really knew they were clicking and interacting with it. And so this local chat would also help make sure that the design parameters, the number of moths, number of robins, and other stuff was actually being trackable and how it actually works. And so if you're looking up at the screen, and hopefully the camera angles have caught this, a bunch of moths were resed, then a bunch of robins were also resed, and the robins are moving around, and they're colliding in their kind of random movement pattern and occasionally colliding with the moth. And then in this case, the chance of death is equal. Now, one thing you can also do real quick is if you zoom in on that little sun globe, that has live text of how much time has elapsed, the number of moths, and the number of births and deaths. And what I'm going to do is pause this really quickly and change the bark color to brown. And so now if you're looking at that disk, depending on what your graphic settings are, it's probably harder for you to see the brown moths compared to the black moths. I mean, that should be a relatively obvious difference. And then over time, eventually we'll see the brown moths take over the population. The black moths will start to disappear. Now, again, I didn't res enough to make this a very obvious and easy thing to occur during the time we have allotted. But this installation is present on Genome Island. Dodge Threebeards is also hosting a version of it at Nova Community College. And so you can play around with this as much as you want. What I'll also do is, into local chat, I will give you the link for the worksheet. So that is available if you want to do that worksheet yourself. Or if you want to adapt the worksheet, talk to me if you want to customize it for your own classrooms. So now if you look up, you'll notice that there are now only 3 black moths, and most of the moths are bound. And this is one of the things that was a very interesting interaction thing to do with the students, is the ability to pause this, right? So when you ask the students to pause it and count something up for the worksheet, and I don't know why this took me so long from the first iterations, but I had to make sure that the time to count it, you put it onto a nice neutral gray background, a very light gray, so that both the moths' color types are easily countable at that particular time. Okay, so I'm going to unpause that. And the other thing to do here too is if you want to teach a more advanced class, I clicked on the sun. If you zoom in on the sun, you'll actually see more information and data about what's actually happening dynamically. So it includes the actual allele frequencies. So if you don't want to have this, so someone else clicked it, but I'll click it again, that if your purpose is not to make students count moths themselves, then you can basically have them record what's going on that sun in order to actually view the actual raw data if you're doing something more complicated, more mathy, and not trying to make them go through the exercise. But that was really, to me, the key thing of trying to build this, is that not only is it a simulator that can help them generate raw data, it's actually putting them in the experience of what the predators is kind of like, of how do I'm looking at this, and the same visual biological concept of camouflage is something they're seeing in Second Life in real time. And so that's my last demonstration. No problem, Lynn. So to summarize, I really wanted to teach, to leverage the Second Life environment, the fact that it's interactive, it's programmable, you have movement, to leverage that and to give the students a really engaging experience, something that hopefully they could take the impression home with them about biology being fun, and also the platform being fun for what they were doing. And also to make Merrill and the people who gave us the grant happy. I think that's a part of it too. But let me just say, it was a lot of extra work. It was so much extra work. And the amount of times I just had to rebuild textures or to recopy textures, or all that work was just it was an incredible amount of work that I had to do, a lot of late nights or weekends. But I will say that overall, I think I got overall good feedback from the students. I think they enjoyed it. There are certainly some students who dropped out and were frustrated and they didn't like it. But the students who stuck with it, the students who were willing to put in the time and just understand that there's a process, that it was a really big payoff. And so with that, I'm happy to take any questions. And really all this stuff I'm happy to pass on and help other people use in their classes. And so thank you for your attention, and thank you for coming.
And thank you, Stephen, immensely. And you never told me that a bunny died. If there are any questions, we have a few minutes here. Please feel free to put them in chat, or you can, we're in voice. You can use voice if you wish. I did not have any questions that were asked during the presentation.
Well, I see a question.
The one question I wanted to ask, you already answered by asking how students were reacting to this. Sort of know. I wanted him to just say it out in public.
Yeah, I still want a backup CD or DVD somewhere. I have the student evaluations, and one of these days I might have to go back and pull that out. So no, I think it was well received. I don't think I know that there were some students who came to later classes that had been told about the class by earlier students, so that's always a good sign. I see a question from Azin Min in the local chat, and asks, how did I get my students into SL, or did they just watch? And the way it worked was students get the ability to register for classes early, like near a month before the end of the previous semester. And you're supposed to have your syllabus and all of the technical requirements and everything listed on the Blackboard page. And so I basically put in as many tutorials as I could for that. I actually made my own video recordings of how to use stuff. I gave them I think, relatively well-detailed worksheets. I also hosted at least 3 workshops ahead of the semester where the students could come into the student computer labs and we would get them set up. And definitely some students took advantage of that. The downside was that there were always students who signed up that day of class, right? You can actually sign up for a class. At the time, you could sign up for a class 5 days after the class had started and still be a full-time registered student for the class. And so that was always the challenge. It was actually the third, I think it was the third time I did this. You know, there was a student who asked me in local chat, "Are you sure you know what you're doing?" Because, right, 'cause I was looking a bit like going on, but what that student didn't realize is that I was answering local chat questions. I was on the phone with a different student. I was doing a Skype conversation with yet another student, and I was also trying to teleport students into the classroom. And so the answer was yes, I did know what I was doing, but it probably did not look like it to her.
I could say that Stephen modeled the potential of SL in his work, and it was a great source of pride for us at the university. Are there other questions? Okay, let me give Stephen a hearty thank you for his presentation. You are invited together informally on the deck at the back of the venue to ask additional questions or to offer comments. Please note that the next installment of the VWEC Expert Series will be on Friday, April 14th at 1 o'clock in the afternoon, SLT. We will host Max Chatnoir, who will speak to us about scripting for educators. There will be no other March meeting in recognition of the VWB PE activities that are going on at the same time. We will post links as needed so you can reach this material and you can see this presentation at other times. And so for now, let me wish you a very good day. And once again, thank you, Stephen. This was exciting. And let me encourage you to join us at the back deck and share with us your thoughts and any other questions that you may have. Oh, wait a minute, Ellie just gave me another question. Oh, I see. No, I didn't see that. Since this time, would you add any new, different generative activities, Stephen?